So we're looking at genetics, and one of the first things that comes up when we think about genetics is DNA. And DNA we tend to draw like this. And it is made up of 4 bases, kind of like its own DNA alphabet, and they are A, T, C, G, and they stand for different molecules. A for adenosine, T for thymine, C for cytosine, and G for guanine. And the way that these get organized down the side of the DNA strand is what tells us all of the information about humans and about other species and even amoebas. And what we find is the way that they connect to each other on when the strand is connected is that they make pairs. So if there's an A over here, it has to connect to a T over here. And if there is a C right here, it has to connect to a G. And we'd find the opposite to be true too. So if we saw a G, it has to connect to a C. And if we saw a T, it has to connect to an A. So these are going to be in pairs. So that's kind of the first thing we need to know about DNA. Now, the next thing we need to know is that when we have a section of DNA and we're looking at it, that section is going to tell us about genes and about heredity, which is really what your class is about. And so we're going to really focus about that information. So if we were to zoom in on a section of our DNA strand, we would be able to see a stretch of DNA, and then in that stretch there would be a section that would be coding for proteins, and that is called the gene. So the gene tells us how to make proteins. And that is the key around genes that we're really focusing on. And Gregory Mendel had a slightly different definition because it was quite a long time ago. And the way that he thought about it was that genes were little independent units of heredity. So really similar, but we've kind of taken it a step further to really think about it in a more DNA focused direction. Now, Gregory Mendel has been coined as like the father of genetics, and he was a monk and he grew lots and lots of pea plants, and he discovered that sometimes he had green plants and sometimes he had yellow peas and sometimes he had white flowers and then purple flowers and wrinkled peas and round peas, and he wanted to know why. And so he came up with this way of thinking about it where he was trying to kind of really decipher what was happening and why. So if he took 2 plants, one of them had green peas and one had yellow peas. Using that information, he started kind of extrapolating. So these are the parents, and so he labeled these as the parents. And then when these were mated to each other, they made a set of offspring. And so our first level of offspring, he called the first filial, and he labeled it as F1. And then those offspring, which we'll get to in a second, made more offspring, and he called those the second filial, and he labeled those F2. What he really wanted to know, though, out of all of this, is how do I know what my offspring are going to look like? And then from there, how do I know what those offspring are going to look like? So that's really what he was trying to figure out throughout this process. I'm going to skip the definitions because you have them in your textbook, so we can just really focus on the Punnett square. But I do recommend that you look over all of the definitions in there so that you know what all of those different key terms mean. All right, so if we look at our yellow and green peas, he saw that there was something happening when we crossed a yellow plant and a green plant. So he looked and he saw that when he had pure or true breed yellow, that meant that there was a likely scenario of having this kind of combination of alleles, basically a way to kind of organize the traits. So he saw that true yellow plants had a dominant yellow trait and a dominant yellow trait, and he saw for green that it had to be two recessive little y's instead. So he was like, well, if I have these two plants, how can I figure out what my offspring are? And so he made a Punnett square. And you can choose which one goes on top and which one goes on the side, but I'm going to just go with our yellow on top. And so I'm going to put them on top just like that, and then our green are on the side, and then they're going to combine. So we're going to put y with little y right here because of these, and then next to it we have a big Y and a little y. Down below we also have a big Y and a little y, and over here there's a Y and a little y. Now, thinking about dominant and recessive genes, if we're seeing a capital Y next to a little y, that color is going to be yellow because we have a capital Y that's dominant compared to the recessive green. So in this case, we have 4 out of 4 are all yellow, and so we have a 100% chance of having yellow peas. So that means all of our offspring are looking the same. Okay, so we have 4 of these in this case. All right, so now what we want to know is what happens if we mate 2 of those offspring, and what would we get in our second filial? So we're going to make a second Punnett square, but using our offspring, or F1. So if we do that, one of our offspring, capital Y, lowercase y, and another capital Y, lowercase y. So in the first box, we have capital and capital. In the right box, we have a capital and a lowercase. In the left bottom, we have a capital and a lowercase. And in the far right one, we have 2 lowercase. So we have a little bit more variability happening. We have 2 capitals, so 2 dominant, and that's definitely going to be yellow, and that's 1 out of 4 boxes. We have capital and lowercase, and that's 2 out of 4 boxes, which is also a half. And then we have little y and little y, which is 1 out of 4 boxes. So we have yellow, and then for the second one, because there's a capital Y there, it's another yellow. And then we have 1 green one because we have 2 recessive. So this is going to tell us what we're expecting for each of our offspring as we keep going, and we could take then 2 of these offspring and put them into a Punnett square and see what happens. And it's a way to kind of figure out exactly how our offspring are going to look going forward and see what kind of ratios and probabilities we should get and expect.